The overall goal of this procedure is to measure proteasome activity in living cells using a GFP-dgn fusion protein. First, clone the custom oligonucleotides for degron and degron frameshift into the PEG-FP-C1 vector and continue with the protocols for the gateway system in Vitrogen. Then produce viruses for each construct using HEK293FT cells. After determining the titer, transduce cells with the virus and apply antibiotic selection. Then expand the resulting stable GFP-dgn or GFP-dgn frameshift expressing cells. This technique facilitates experiments that monitor cell responses to given treatments through fluorescence signal measurements. For instance, proteasome activity can be easily assessed using epifluorescence or flow cytometry.
The main advantage of this technique over existing methods like luminogenic or fluorogenic assays using whole cell lysates is that the proteasome activity is measured in living cells.
For the transfection, dilute 36 microliters of lipofectamine 2000 reagent in 1.5 milliliters of DMEM. In another 15 milliliter Falcum tube, dilute the DNA constructs in 1.5 milliliters of DMEM. After 5 minutes, combine the two and incubate for 20 minutes at room temperature. Now, replace the medium from a confluent culture of